Next, I want to show you the example files. So this is what you'll see if you download the example project. There's a paper, the paper that these images came from, and there's 4 images. Now, if we actually look at the paper itself, what we can find is that it's RIF1 supports the function of the CST complex in these telomere cabin. And if we look down to figure 2, we see the images that this came from. So essentially, this is a senescence study. There's different time points. So generation 1, 2, 3, and 4. And then there's 4 different genotypes. So we have 4 time-based variables. So this is your ordinal variable. And then you have 4 categories. So these are genotypes. So in the context of QuantaColony, projects can contain ordinal variables, nominal variables, or both. That'll make a little bit more sense as I get into it. But in this case, we know that the ordinal variable is the generation number of 1, 2, 3, and 4. And the nominal variable is these genotypes. Anyway, so I have those 4 images and I have a PDF. We actually get started. I would double click on that QuantaColony shortcut to get started. On first opening, this may be pretty slow, so give it about a minute. If the software is a little slow to start. But I end up with this page. This is how I know that it works. And this is the project selection screen. Here is where I can select my project, and I'm going to create a new project. I can do new projects. Here, I'm choosing the files that I will be using for my analysis, the input files. So that's going to select files, and I can maneuver to my desktop. These are the images. I can select them all, hit open. And these are the files, example gen. So I can rename the project however I want. Let's just call it, project 1. Okay, and we can create the projects. Now, QuantaColony is going to ask us various configuration questions associated with our projects. First of all, what plates are we using? There are rectangular plates and there are circular plates. Both are supported. This is an example of circular plates. And then it's going to ask us how many sections our Petri dish is broken into. In this case, it's actually 4 sections. Okay. Now, we said that our data has 2 variables, as the nominal, which is genotypes, and as the ordinal data, which is generation number, which is why we would use both. Now, if you had like a series of just one mutant across 4 generations, or maybe a temperature series, or maybe a drug concentration series, and you only have that one category, you would choose ordinal only. And for nominal only, you would have maybe 4 genotypes at one condition. So in this case, we have both types. And then it's going to ask me how my plates are organized. So is my nominal variable, my genotypes, are those organized in the sections of the plates, or the genotypes organized across the different plates? Or alternatively, some other weird thing that is not described by those 2. Like maybe you have some pictures with some genotypes and some pictures with other genotypes. So complex is for anything else. So you have your standard layout. And there's actually a help dialog here you can use if you're confused. Okay, now it's asking me how my plates are organized. So there, I said that there were 4 sections: top left, top right, bottom left, bottom right. Now it's also going to ask me what all these sections represent. So I said that these were genotypes. I could have chosen species or maybe something else where a dialog will open up for me to list what these categories represent. These are genotypes. And if we go back to the original picture, we can see that the top left was a double mutants. Top right was CDC13-2, bottom left was wild type, bottom right was a RIF1 deletion. So I'm actually going to list that here. So we said double mutant over the top left. We have top right CDC13-2. We have bottom left wild type. And then we have bottom right RIF1 deletion. I'm going to now list which of the 4 is my control group. So these checkboxes indicate which of these are my control. Wild type is the control group. So continue. You have to choose a control for QuantaColony. Continue. Next, now this is asking me how my ordinal variables are organized. Remember that I uploaded 4 separate photos. So those photos in this case are the ordinal variable, right? We have photo 1, 2, 3, and 4. So QuantaColony is asking me, what do those represent? Well, they represent different generations, as we said. So this automatically loads to be the generation number. And it's asking me which is the control generation. That'd be generation 1. Now, there's something I can do here. I can reorder these. Let's say that I uploaded the pictures in the wrong order. I can, for example, so it's saying that this photo, Gen 2, should correspond to Gen 2. But let's say for some reason that's wrong. I can actually click and move down, and you see that swaps. So, but that's, we had it correct in the first place. So this is assigning the photos that I uploaded to the ordinal variable designations that we're choosing here. Okay. So now we've essentially told the program our nominals are in sections, our ordinals are in different photos. We can continue. It's going to ask us how scaling works. So essentially, there's 3 different ways QuantaColony supports conversion factors for scaling for real-world units. You can use plate dimensions, so the size of the Petri dish as a whole. If you have an image that has a scale bar in the image, we can use that alternatively. Or if you don't want to use any conversion factor at all, you can use no scaling needed. Okay, so I'm going to use plate dimensions. And it's asking me what is the size of my petri dish in radius. Now the standard petri dish size is 4.5 centimeters, so I have that here and hit continue. Okay, so now we have project one, and we can open this and we can get started. Let's open the project. This is actually the page where you're going to select projects. This is sort of the very starts. You can combine projects here. You can export projects. You can do a lot of stuff here. So let's get this started.